I want to start with thanking you firstly for being with us here today. It is a very busy year. You've heard the UN Special Envoy for the Ocean, Ambassador Peter Thompson, say many times that this is the year when we're going to stop the decline of the ocean health. The Bbnj agreement offers a very significant part as one of the major pillars on how can we ensure, that biodiversity is both conserved and sustainable use in the high seas. So it's a very, very important and urgent task that we have in front of us. So IUCN has been engaged in this process, and we have started a knowledge series for in 2020 in order to keep the momentum going, as well as the political will and being a resource. And this is something that we do in collaboration with others. So we're very pleased today to actually be joining forces with the European Commission, but also with the support of Marbles and the Deep Sea Ocean Stewardship Initiative. So with that, today we are going to hear about, the marine genetic resources and to be exploring scientific, practical, and wider uses of conservation within the context of the Bbnj agreement. So we will hear about, we will have the privilege of listening to experts, ambassadors, and other distinguished speakers to talk about, the importance of marine genetic resources and the very need to advance the knowledge, both scientific, but also the traditional and ecological knowledge, and to look at what are the data needs, how to go from data collection and to data management, and also applying the fair data principles. So we're looking forward to that. And I would really like to end by thanking not just the organizers and the collaborator, but really all the delegates, because you're the ones doing the hard work. We're in the negotiation room, so we really, on behalf of the ocean community, want to thank each and every delegate for all the work that you do, and both in the intersessional period and at these negotiations. And you have a very important task and a challenging task ahead of you to strike that balance between to get to where we want to be in terms of international collaboration. And I believe now is the time that we need multilateralism, international collaboration more than ever. So once again, thank you very much. And I wish you a very good webinar. This webinar will be recorded, and it will be shared on IUCN YouTube channel, so it can be revisited later. So with that, I wish you a good webinar, and I'll hand it back. Thank you very much, and I look forward to seeing you in New York while we make history together. Thank you.
Oops. Okay, so I hope today it worked. Sorry for the hiccup of yesterday. I'm not super good at the technology. So thanks again to IUCN for this message, to Mina, and for sharing this message with us. So as I said, we would like also to thank all the participants of yesterday, and also all the experts that intervened yesterday. We had a very interesting webinar, which lasted even 20 minutes more than expected. We were running a bit out of time. And so let's keep going. Today, I will introduce the main messages that we received from yesterday. So let me just project my presentation. Okay. Can you see my screen? Can somebody say something? Does it work?
Yes.
Okay, thank you very much. So yes, I would like to introduce the main messages we got from the expert that intervened yesterday. As not being a scientist in marine biology, I will do my best to respect their languages. And I also ask permissions to the, I also ask, sorry, do you see it in full screen? Yes, now better. I also ask
Not
permission.
yet.
Not yet? No?
Yes. Yes.
Okay, thank you, Christina. As I said, I also asked permission to some of the experts to use directly their slide, to use directly their language. So thanks, everyone. So in the first panel, we went around the oceans with the important research projects, and we looked at the importance of MGR for conservation and advancing knowledge of the oceans. So we saw we were driven by Tara Ocean Expedition, by the iAtlantic project, and by some research done in the Pacific on coral by the Cryob Center. And we asked the scientists after what is the message they would like to deliver to Bbnj. What could Bbnj do for science? And the main messages were that the Bbnj agreement should ensure that ABNJ remain open to scientific exploration for basic research, and the DSI derived should remain open to all for the benefit of humankind to enhance knowledge. Another message reflects the need to protect coral reefs and other biodiversity hotspots, in which in most of the cases are still to be discovered. And another message regarded the acknowledgement of the imbalance in knowledge in relation to the different regions of the oceans. And one example was brought by this Global Ocean Science report on the difference in studies effort, for example, in comparison from the North Atlantic to the South Atlantic. So this is also a message that should be linked maybe with the capacity building panel that we have today. And so we can look also in the bottom where we see that there are very little records in the ABNJ for coral reefs. So the message is that many hotspots are still to be discovered. So there's a lot of work to do by science and by policymakers to protect what we still don't know what is there. From panel 2, we wrote with the Convention on Biological Diversity Secretariat. So we invited them to deliver a presentation on the policy option for benefit sharing for DSI that are under discussion in the Convention on Biological Diversity. And we invited also to ensure more synergy between the two different negotiating framework, because we believe that biodiversity is one and we should treat it as such, and we should ensure coherence and consistency between different framework that deals with biodiversity. So they deliver some very important information from the recent, from the third meeting of the working group, which agreed on certain recommendation to be discussed or also again next week, I think, and to be delivered to the COP 15, which for which we don't really know a date and the setting for the moment. But this recommendation regarded a solution on benefit sharing for DSI, and the parameters where they see a solution for the moment is that this solution should be efficient, feasible, and practical, should generate more benefits than costs, so be effective, should provide legal certainty for providers and users of DSI, and should not hinder research and innovation, and be consistent with open access to data. Then they also analyzed some of the point of convergence from the advisory group on DSI, and some element of this convergence is that tracking and trace could be used for a limited specialized subset of DSI, but is currently not feasible technically or financially at a large scale. And the solution of DSI is likely at the moment to include a multilateral mechanism. So, and there was another very powerful message, we think, from panels 2. So the need to ensure synergies between the two different negotiations and to find a global solution because simply biodiversity as a whole is in an extreme state of crisis. Then we went to panel 3, where we had also here excellent expert driving us from collection to data, explaining what is a unique identifier and elaborating on the production of data. This is a slide I give credit to Muriel Rabon. Thank you. And these are her takeaway and her messages to the audience after the presentation. So she highlighted that existing framework for persistent unique identifiers are already existing in science, and they do allow for traceability of marine genetic resources. And they are also in line with, they also allow open access and the FAIR principle. So basically, the suggestion is to support and recognize these existing framework, which are based on persistent unique identifiers, and to adapt with them. So the requirements for additional reporting or potentially invalid identifier might potentially create confusion and add administrative burden to science. So the suggestion was more to rely on best scientific practices than to elaborate on alternative identifier, which would not be unique probably, or would not be interoperable within each other, and would not respect the condition that the scientific persistent unique identifier respect. From the point of view of commercial research, she highlighted that there is a different pattern. Sometimes it's a more fragmented and more difficult to access certain information. So the private sector could benefit from being more transparent and more in view of open access. Oops, sorry. And the last message is that it's essential not to create unworkable system or require burdensome administrative steps, particularly for public sector science, which usually operates within limited budget and also without access to legal teams, something that I never considered, but was an additional input I got yesterday. From panel 4, we dived into databases and management and use of data. So from Guy Cochran, I took these slides where we see the enormous amount of data that are produced. For example, one new data set every 6 minutes. And these are just the number of the amount of data that is produced and available in open access in the INSDC. And from Amber Schultz, we learned that the Bbnj DSI dataset is quite small, is approximately 1% of the whole databases. And I will come to that when she highlights her suggestion for the Bbnj. In the view of Amber Schultz and also a group of experts who participate in the DSI scientific network, they elaborated on 5 principles of a win-win DSI benefit sharing solution, which should be based on open access, simplicity, should harmonize different frameworks, and should recognize the goal of conservation and sustainable use of biodiversity, and should also be fair and transparent. And her suggestion for the Bbnj community is to recognize that MGR DSI, as spotted, is quite limited as a dataset compared to the whole dataset, and also to consider the unintended consequence of creating a system that can disincentivize the use of this system, in the sense that if the Bbnj MGR regime would end up being more restrictive than the CBD regime, for example, we could expect users to avoid the MGR Bbnj regime. So this is another reason why harmonization should be ensured between the different international regimes. And of course, a recognition that inequality exists in terms of capacity building for DSI and MGR utilization. And this is a slide that has been used both by the CBD Secretariat and which shows the different 6 options under discussion for benefit sharing for DSI. And the suggestion of this group of experts is to look into option 3, 2, and 4 rather than the other. I project this slide, which might seem a bit overwhelming now, just to prepare the discussion maybe in panel 7, where we have all the experts coming back and answering to our questions. So I hope we will have lots of interesting interaction. Thank you very much. And okay, I'll stop sharing. And I would like to leave the floor to Ambassador Rina Lee. She doesn't need introduction. She is the president of Intergovernmental Conference for Bbnj, and we are very glad to have her here with us. So the floor is yours, Rina.
Thank you very much, Ariana. Good morning, good afternoon, and good evening to everyone in this webinar, wherever you may be. Very happy to be here, and first of all, want to thank the organizers of this webinar, the European Commission, IUCN, the Marbles Project, and DOSI for inviting me to join you in this webinar. I'm sorry I couldn't join you yesterday. The time zone differences did not work out for me, but I'm very happy to be here today. I guess by now, some, if not all of you, would have at least taken a cursory look at the further revised text that has been issued. I hope that you can, I mean, this is not a forum for going into the details of the revised draft text, but I hope that you can begin to see for the topics that we are discussing today, MGRs as well as capacity building, you can see the sort of framework, or you can have a look at what we're trying to do, what the future may look like. And to say that, as I did in the 4th session, that in order to be able to future-proof this agreement, we do need to remember that we are looking at setting out directions, but not details. Put in another way, what we're trying to do is to build a scaffold in the agreement, so that as we know more and we learn more, we understand more, we can then begin to, we can flesh out the scaffolding, and we can have the flexibility to adjust as the years go by, because we do want this to last for many, many years to come. And listening to Ariana's presentation on the points that were made yesterday, particularly also the need to have some degree of coherence with the work of CBD, the CBD in terms of DSI, a scaffold makes sense because it gives you that pathway to be able to kind of absorb some of the lessons or some of the practices that are being negotiated in that forum. And I also, of course, I think what the other thing we have in common is that I think we're both advocating practical solutions. What we want to do is to build an agreement that is implementable. That means that the solutions, the mechanisms, the processes, they must be practical. They must be implementable, for want of a better word. So I think it's important for us to bear this in mind. And when I looked at the program, when the commission first reached out to me and I looked at the program, I didn't recognize the names at all in the program. other than the moderators. I did not recognize the names, which is to me a very good thing, because it's really important for the Bbnj IGC process to be able to hear the views of those who may not be participating directly in the process, or who, even though they may be advising delegations, don't always get a chance to speak in the rooms. As you know, we tend to prioritize delegations, member state delegations, and sometimes you may not. And of course, I don't need to repeat what restrictions there were for our fall session. And it's opportunities or instances or platforms like this webinar that allows us to hear from people who, although not directly involved in the process, may be or are likely to be affected by what it is we're trying to build in that agreement, or who have to subsequently implement the rules that we are writing. So even though for some people they may not be able to participate directly, not everybody can always have their views heard directly, but giving that channel, that voice, I think is very important. And I think this sense of inclusivity and ability for everyone to be engaged, to be involved in whatever final mechanisms, processes that we build and we agree on in the agreement, I think is an important element of building a fair, balanced, and most importantly, an effective agreement. And also reminds us, particularly those of us who are, who are deep in the throes of negotiating the agreement. It reminds us that this is not just a legal endeavor, that we must look at the complex issues, not just from a legal perspective, but also from a policy perspective, as well as a scientific perspective. So being able to bring the policymakers, the lawyers, and the scientists together, I think it's very helpful, because what will help us to achieve our objective of conservation and sustainable use is the ability to take science-based decisions, but also, and understanding the role that may be played by traditional knowledge as well. So I'm not going to take up too much of your time. I'm sure everybody is looking forward to listening to our speakers today. Once again, to thank everyone and to thank the organizers, and to wish everyone a very good webinar. I really look forward to learning from our speakers today, and to say, maybe one final remark, which is that I know many of us in the audience are probably gearing up and making our preparations for the Ocean Conference. And I guess I'll probably see some, if not all of you, in Lisbon for the Ocean Conference. And it's a reminder that ours is not, the Bbnj IGC process is not the only process out there, that there are other processes, other programs that are also out there with I guess a shared ideal of wanting to be able to contribute to the conservation, to the sustainable use of the areas beyond national jurisdiction. So it's important for us to see our work in that context, and to understand how we will operate in that broader context. And with this, I'm going to wish everybody a good webinar, and I want to thank in advance all the speakers. Also, I look forward to learning from you. Thank you very much. Thank you.
Thank you very much, Ambassador Rina. Thanks. We are glad you're here. So now we can go to panel 5, which is titled Example of Discovery Processes. And I will introduce the moderator, which is Frank Amfires. Dr. Frank Amfire is a legal lecturer at Queensland University of Technology in Australia, Over the past 20 years, she has worked in government, academia, and as a consultant in fields of marine law and policy, including biodiversity, biotechnology, fisheries, and maritime law. She has been on the International Council on Environmental Laws delegation since the start of a negotiation for the proposed internationally legally binding instrument on the conservation and sustainable use of marine biodiversity in areas beyond national jurisdiction. So I leave the floor to Fran, and I anticipate that we have only 2 speakers in this panel because the third one, unfortunately, is sick, so we don't have Joape Gini with us. It's a pity because we reached out to the Pacific scientific community and we were very glad to have him on board, but unfortunately he is sick. Fran, the floor is yours. Thank you.
Thank you, Ariana. I acknowledge the traditional owners of the lands where I'm speaking from, and I respect their elders, past, present, and future. And I have the great pleasure of introducing our first speaker, Ibon Cancio, who's an associate professor with the Zoology and Animal Cell Biology Department at the University of Basque Country. He's a researcher in the Consolidated Research Group in Cell Biology and Environmental Toxicology of the Basque Government. He's responsible for the incorporation of the Plentia Marine Station to the European Marine Biology Resource Centre. His research activities focus on 2 main interrelated research lines: cell and molecular biology of fish sex differentiation and applications of cell biology in toxicology. He's here to give us some wonderful insights into how scientists use the information in the nucleotide sequence databases. So please welcome Ibon.
So hello everybody. So I hope I'm able to share my presentation right now. Here it is. Can everybody see it?
Yes.
So thanks for the introduction, thanks for the invitation. Above everything that it has been said, I am a professor and my job is to teach people. And I have been invited here to teach you on DSI. So I'm going to take you through a walk through the wild side, through the wild side of DSI. And I'm going to show you how we scientists use digital sequence information available in the nucleotide sequence databases. So this is the place where I work. This is my marine biological station. And we do research and we do provide research services on biological resources. Some of them, we collect them in the field. Some of them, they are in culture collections. Some of them are in biobanks. Some of them, they fit on the scope of the Nagoya Protocol. Some of them come from areas beyond national jurisdiction. And when working with these genetic resources, we produce data resources coming from those genetic resources, which at the moment, they are not within the ABS scope. We produce digital sequence information and contextual data, and we share this data. So for the people listening that may be not understanding, what is digital sequence information to some extent? So how is information of life coded? It's very easy to understand how information, feeling, being in English is coded. It's coded by 26 letters. And you put these 26 letters into different orders and they produce amazing things like to be or not to be. That is the question. It's a piece of information that is kept in an organism that is called Hamlet. So how is life coded? So 4 nucleotides, 4 different letters, A, T, C, and G, code for any genetic resource. And the differences between different species are coded in this digital sequence information. So how do we researchers do work with this different language? Myself, I go to the field. In my case, mostly it's coastal genetic resources that fall under the sovereignty of one country, mainly Spain. I access these resources. I do utilize these genetic resources, and in many circumstances, I do sequence the information in the DNA of these resources. And as a scientist, I need to share this information with all my colleagues. And to share this information, I do input this sequence information, this digital sequence information, into the ENSDC, the International Nucleotide Sequence Database Collaboration. This International Nucleotide Sequence Database Collaboration, it's placed in 3 mirror places. There are 3 major databases, public, freely available databases, one in the United States, one in Japan, and the other one in Europe. And this, it's like the big library of life, digitalized library of life, that as a scientist, I can access to get information. So now, I'm going to do the teacher job, and I'm going to do some exercises with you. We have 3 exercises. The first one, the 3 ENSDC databases are interconnected. They use unique identifiers. And we are going to be doing an exercise, mining a piece of text, and we are going to be looking for a sequence search. And I will be showing you how these unique identifiers are really true. This is going to be the first exercise. Then I will show you how DSI from all genetic resources in planet Earth is interconnected. This big library, it's a big puzzle. And if you take one of the pieces within the puzzle falls apart. And for that, we will have an exercise. The third exercise will be, okay, I have a sequence that I have found in the environment, and I can search for this sequence, and the databases will tell me which species this sequence belongs to. So I can use this digital sequence information for biodiversity monitoring. And 4, it's important to understand that ENSDC is deeply interconnected with other databases, with hundreds of databases. And through exercises 1, 2, and 3, I will be able to show you this, I hope. So first exercise, let's look for a sequence of interest in the ENSDC database. So the chimpanzee is an octocoral, a colonial cnidarian, and you know that many marine organisms, they emit light, they emit bioluminescence. And they do that through a gene that codes for a protein that is called luciferase. The name comes from Lucifer, which is the light, the one that brings the light. And this gene that produces this enzyme is very much used in biomedicine because the protein that it produces light, and it allows us to check in different cell systems where different processes are happening because we are able to see this light. So let's go to the first exercise. For that, I'm going to be going to the databases, and this is the first database. This is the National Library of Medicine in the United States, and I'm going to be writing Rhenilla reniformis luciferase. Rhenilla reniformis is the chimpanzee. And when first, here it says all databases, I need to select for nucleotide. I write Rhenilla luciferase. Rhenilla luciferase. Reniformis luciferase. I search. And it gives me a series of Rhenilla reniformis, but if you see the first one, Rhenilla reniformis luciferase mRNA complete CDS, it tells you an accession code, M63501. This defines this sequence in the database. Yeah. Now I'm going to be going to the INA browser. This is the one in the European Nucleotide Archive. I can search and I can write Rhenilla reniformis luciferase. I'm going to go here. In the European Nucleotide Archive, I have a sequence for Rhenilla reniformis luciferase, which is code M63501. So the same sequence is identified with the same unique identifier in the American and in the European Nucleotide Archive. Let's go now to the Japanese. And in the Japanese one, if I search for the DNA information, search by keywords. I look for Rhenilla luciferase, and it gives me a series of sequences, and among them you find Rhenilla reniformis luciferase with the accession code M63501, exactly the same as the others before. If we come back here to the American one, we can click on the Rhenilla reniformis luciferase, and you have the information of the sequence, the protein sequence that is derived from this nucleotide information, and you find the name of the authors of this sequence that publish a paper that, as it is interconnected, we can click and we can have a look at the paper in which this sequence was published. It was published by Lawrence et al., and you can go into the paper and you will find that this chimpanzee, this marine organism, was obtained from the coastal waters of Georgia in the United States. Another thing that you can do is this sequence, you can check for similar sequences within the database. For that, you run a blast. Blast is like writing to be or not to be in Google to find which is the book in which it is contained. So if you run a blast, here it uses the unique identifier to compare with all the database, with all the information in the database. If we do this analysis, okay, it will take a little while. So first, I already did the analysis beforehand, and you can find here that Rhenilla reniformis, my sequence, is very similar to Rhenilla reniformis luciferase. Of course, it's the same sequence, but the rest of the sequences that are similar to Rhenilla reniformis luciferase are expression vectors, cloning vectors. Those are vectors that have been produced in the lab, incorporating the Rhenilla luciferase in order to use them in different cell biological approaches. Good. Now, let's move to the second exercise. Let us search for a sequence. Okay, I have been working with this sea anemone, Anemona viridis, which is a species of anemona, very similar to corals, but it doesn't calcify. It has got a symbiont inside, and it can suffer bleaching. It can expel the symbiont. This is the problem that we are facing with coral bleaching in the tropical areas. We don't have these corals, but we have sea anemones. And I am interested in studying this bleaching effect. Okay, this sequence I found in these anemones, in these sea anemones, and I am interested to know what gene does it belong to, which is the function of this gene. So I can take the sequence, I can come to the database, and I can blast the nucleotide sequence. That means I can write here the sequence, I can write to be or not to be, and then I can interrogate the whole database about who does this sequence belong to. Yeah. Okay, I have done this in advance because this takes a little while to run. And you see, it tells me that my Anemona viridis sequence is very similar to all the sequences here. And all the sequences belong to a gene that is called glutathione reductase. This is an antioxidant enzyme, and oxidative stress is very important when bleaching is happening. And the species that appear here, Actinia tenebrosa, Exaptasia pallida, Nematostella bectensis, Stylotella pistillata, these are also cnidarians like my anemona, and some of them are corals. But some of the species, for instance, Mercenaria mercenaria, is a mollusk, it's a bivalve. Rana temporaria, it's a frog. Orbiomphalaria glabrata, it's a snail. Those are species in which there is a glutathione reductase very similar to my sequence of interest. So now I know that I have glutathione reductase. And what is Actinia tenebrosa? So I can go to the database. Sorry, I need to. If I go to GBIF, I'm going to put it in English. I can write Actinia tenebrosa here. I check for Actinia tenebrosa, and this is Actinia tenebrosa, and I find the place where this Actinia tenebrosa lives. So Actinia tenebrosa is a species from Oceania. My species, Anemona viridis, it's a species that I have collected in Spain, and it's typical in Europe and in the north coast of Africa. But my sequence of glutathione reductase, I have been able to identify through comparison with the sequence of a species that is in Oceania. If the sequence of this species in Oceania wouldn't be available in the database, it would have been very difficult to identify my sequence in my species collected in Spain. Good. Now, let's go to the last example. So the last example, within ENBSC, we are establishing an oceanic genomic observatory in which we are addressing ABS compliance, and we are seeking to facilitate the due diligence from sampling to data. And what we do is we collect water samples in 16 different places in Europe and in the Red Sea. And this is the place where I collect my water sample. Everybody knows the biggest city in the world, which is Bilbao. And in this biggest city in the world, every 2 months we collect a water sample and we sequence. And this is a sequence that we obtain. Oh, sorry, that we did obtain. And we do not know this sequence. It belongs to a mitochondrial gene, but we do not know to which species does it belong. So if we take the sequence, we can go to the database and we can blast. We bring it to the database. We interrogate the whole database. And when we interrogate the whole database, this is the information I obtain. My sequence belongs to Pseudodiaptomus marinus. Yeah, all these organisms are Pseudodiaptomus marinus. If I look all the list of organisms that are similar to this sequence, they are crustaceans of the Calanus family. But what is Pseudodiaptomus marinus? If I go to Pseudodiaptomus marinus and I check what it is, this is Pseudodiaptomus marinus. And if you look in the map, it's a species that up to now has been localized in Japan. It's a species originally from Japan that along the years has begun to appear in other places in the United States, in some places in Africa. There is one localization in Europe. Yeah. So with this sequencing approach, I have been able to identify the presence of a species that is originally from Japan, but I have been able to spot its presence within the coastal waters of the Bay of Biscay in the north of the Iberian Peninsula. So this is a very good system to study where different organisms live. To finish, I'm going to leave you with one map. If I check bacteria here, this is the map of bacteria in the world, all the bacteria that have been recognized in the world. And you will see that there is plenty of information of presence of bacteria, different species of bacteria on Earth, but there is plenty of gaps in knowledge in the areas beyond national jurisdiction. And obtaining information, digital sequence information of all these organisms would be a very good way of understanding the biodiversity in planet Earth. So with this, I think I have finished, and I hope I have been able to show you a little bit, as if you were my students, what DSI is about. Thanks a lot.
Thank you so much, Yvonne. That was an incredibly I guess, clear explanation for shedding light on, the language of DNA to help us all feel like scientists. So thank you so much. I think you did a really good job as well, I guess, showing how all of the databases are related and, the continuity of the identifiers that flow between the databases. And I think it was excellent. I guess it may come into our discussion a little bit later in terms of your explanation of how a blast search works. And, when we talk a little bit later about understanding how DSI is used in the sense of access. One other point I think you raised that was really interesting was that you came up with other similar sequences in vectors that are used in the lab using the sequence that you were searching for. And that's very important, I think, to remember that, the resources can be used as a research tool as well as a genetic resource themselves. So thank you. We will have questions after Marcel's presentation. But if anyone has questions anyway, you can put them into the chat and then we can store them up for after Marcel's presentation. So I have the great pleasure of introducing Marcel Jaspaz. His main expertise is in the discovery, characterization, utilization, and biosynthesis of natural products produced by marine and desert microorganisms. This forms the core of the biodiscovery pipeline, and Marcel works with people operating at all stages of this pipeline, from the collection and identification of the organisms to their testing in whole animal models. Marcel directs the interdisciplinary Marine Biodiscovery Centre at the University of Aberdeen, which focuses on microbial resources for novel pharmaceuticals. The centre contains facilities for chemistry, chromatography, spectroscopy, molecular genetics, and microbiology. Marcel has been active at national and international levels to develop the science, its applications, and associated policy involved in biodiscovery and biotechnology. So he's here today to give his astute perspectives on the biodiscovery process with examples of EU projects as best practices for biodiscovery. Thanks, Marcel. Marcel, have you got your microphone on?
Marcel, are you there? Don't see him online anymore.
I can see him on the screen. Oh.
Yes, I see his presentation, but I don't see him anymore as participant. So maybe he had a problem. Oh, he's back.
Oh, there he is. He's on the presentation.
We cannot hear you. Okay.
Sorry,
Perfect.
I think you came out. I couldn't figure out how to unmute myself because as soon as I set the presentation up, everything else disappears. But I found it now. Okay, thank you very much. Thanks, Fran, for your kind introduction, and thank you for the organizers for inviting me here to speak today. And again, Yvonne, thank you very much for a very astute presentation on how DSI is actually used in practice. I wanted to extend that a little bit by talking about European projects, how they work, and how they are able to connect a very long pipeline of scientists in different parts of the world, including companies and policymakers, to try and work out what is best for biodiscovery at an EU and perhaps even global level. So I'll now try and make that work. Yep. So just to say, a lot of these EU calls are made based on reports that are written. So for instance, this report here on the right-hand side is a position paper written by the European Science Foundation in 2010, which led to some of that text being used for a European Union call. And they make it clear that there is 24 million euros available and that you will have more than one consortium funded, and that 25% has to be industry. And it tells you very clearly what's required, right? And again, it asks for a properly integrated effort focusing on industry requirements. So we were lucky to get funded. So we called it the pharmacy project initially. So it is a 9.5 million euro fund that was basically the 24 million was actually increased to 27.5 million, and 3 big bids were funded. In our case, we had 24 partners from, I think, 14 different countries. And we had this sort of mission statement to improve the quality, volume, and value of active agents discovered in the marine environment, and increase the speed at which they can be delivered to the marketplace by addressing bottlenecks and restrictions and adding technical booster plans. It sounded very good at the time. I'll tell you a little bit more. Most of the focus in this case was on deep and cold ocean environments, which meant that we got involved after the first year in the Bbnj process at the stage of the working group, then at the stage of the PrepCom, and later on with the IGC process as well. I was the scientific coordinator, but the project coordination, the administrative coordination was done by the University KU Leuven, first by Camilla and then by Peter. So the idea was that we would look at all the legal and technical barriers to the work we were trying to do. And initially, we figured out that there was important physical access problems to the deep ocean, and that also there were legal access problems. Again, the uncertainty of what could and couldn't be done under UNCLOS and how the process would change over time. And we wanted our scientists to get involved in this process. What we always think is that unusual biodiversity, unusual environments give rise to unusual biodiversity. So we were very clear from the work that we'd done previously that novel biology would come out of this. And we would need to be able to isolate that interesting biology. So take a sediment sample from the sea floor, isolate the bacterium, and that then allows us to identify new chemical talent. And that chemical talent leads to new chemistry. And that requires a lot of chemical investigation. Fran referred to spectroscopy in my background, and that is indeed the techniques we need to use to determine the structure of that. And we use a lot of data mining techniques as well nowadays. So then often with new chemistry comes new biological activity. In this case, for instance, against epilepsy, we again determine whether or not something is toxic or not. And then a long road is taken from activity to product. It can take up to 20 years sometimes. So we promised at the beginning to have 2,500 microbial strains, out of which we would make 18,000 extracts, which would then be filtered to 540 bioactive extracts, about 115 molecular families, and 2 leads that go into animal trials. The reason for this sequence of numbers is because we know from previous work that success rates are low at certain steps, and that therefore we need to be able to have a very large starting base to end up with a product that might have a chance of going further. So here's an example of the process from the Irish EEZ, and they collected with their vessel, with an ROV, using live video sampling. They collected sponges from the deep sea and also sediments. From that, they obtained some beautiful bacteria, and they also identified all that bacteria using the DNA sequencing that Ivan has already mentioned. They were able to build a family tree of all the different strains they found and how they related to each other in the deep sea. So that was very exciting to see that. We also, during the project, developed standard operating procedures. That meant that we're all working towards the same testing of these organisms in biological assays using the same procedures. And that was very helpful to decide that early on, because that allowed us to validate all our results against each other. I just want to make a statement here, because Ivan already referred to it, that some strains are everywhere. And there was a statement made by a Dutch microbiologist in 1934, Lawrence Baas-Becking, and he said, everything is everywhere, but the environment selects. The most important thing to remember then is that many of the species that we may think only belong in one location, in fact, occur in other locations as well. Ivan already mentioned the fact that he found gene sequences that apparently he found in Spain were also present in Australian species. And again, it's important to remember that nothing is really unique globally. You'll find some things that seem new, and then you'll find it's already been found elsewhere. So it's quite interesting to think about that carefully as to how that will affect any of the negotiations you're set to do. So the really important part of these projects is data management. So we have all the different types of data, a huge variety of data. We have essentially the need for a geographic information system. We have the need for some way of identifying bacterial information, chemical information, bioassay information, and then clinical information as well. And we used a database at the time called OpenNaPis, which is a database that allows you to do all of that. So it's essentially a laboratory information management system on top of a geographic information system. This is incredibly complex, and curating such a database was a real challenge, and it took Jeanette a lot of time to try and do this. Given that we had over 130,000 data points, this is just a small project. Having a customized database system to try and track and trace everything is very, very challenging if it's outside of a single big project. Again, if it's multiple projects trying to do this, it would be very challenging to achieve this. So what we heard yesterday from Muriel Rabon, again, was very important to recognize that unique identifiers are important to be able to follow this in an automated way using databases that already exist. In-house databases like this are essential for industry, in effect, to make follow their own data, but good science is built on open data these days, and it's critical that we try and understand that and follow the best practices in science. So we already said many types of different data and disparate types of data with databases at different levels of development as well. So how did we go in pharmacy? Well, we did very well, actually. We ended up with more strains than we said we would. We ended up with fewer active extracts than we said we would. We had about 600 active dereplicated extracts. We got 90 compound families, but we ended up with a success rate of 5 drug leads. 3 against epilepsy and 2 against Alzheimer's disease. So that was quite successful. However, none of these drug leads came from Bbnj. They all came from territorial waters, so within the EEZ. And it's important to remember that there isn't much information yet of microbial natural products from Bbnj, and therefore we have to think very carefully how big the potential is. It's probably quite large, but the question is how many people are really going to look at that given the extreme costs involved in mounting an expedition to Bbnj. The other things that we did in pharmacy was to create a science policy interface which allowed practitioners and legal experts and policymakers to talk together. We had side events at for instance, the COP20 something and the IGCs and the CAPCOMs. We wrote papers for the scientific community. We shared best practice for the scientists, and we even put together a proposal, an early proposal for building blocks towards the solution of the problem of marine genetic resources of areas beyond natural jurisdiction. Most importantly, just to recognize the key author in that was, of course, our very own Ariana Brogiato, who's leading today's meeting. From this, several of the partners got together and founded a core group for another proposal to the EU called the Marbles Consortium, which is harnessing microbes for drug discovery and sustainable production of fish and crops. And we have a huge complementarity of expertise, and we also included European infrastructure such as EU OpenScreen. And this time, the things that we're looking at are the microbes that grow either in sponges, on algae, or on fish. So we're looking at symbiosis, so we're looking at how important that is. So we can see again the bacteria that live together with these things often harmoniously can be of benefit to the organisms themselves to make them live healthy lives. And again, the idea then is that we look at how we can realize the biosynthetic potential. So it's a lot, again, of genome information. So this is a metagenome from a sponge, where you look at an enormous number of bacteria that grow within an organism that all seem to be happily living together, or at least coexisting. And the other side, each of these genes inside these organisms is able to produce a metabolite. And sometimes you don't know what those metabolites are. So we need to try and figure out what the true molecular diversity is in these organisms and how we can access it. So again, this is a challenge that we're trying to address in the Marbles project. You can see then we have several work packages. We have the first 3 work packages really aimed at biodiversity mining and omics-guided technology to try and figure out which of these strains are of interest, how strains can live together, and how combinations of bacterial strains might be useful, for instance, in aquaculture to prevent infections of farmed fish. And that would be using the microbes themselves that already exist in nature together rather than using small molecules. The alternative approach is then, for those where that's not possible, to use small molecule chemistry derived from those organisms to try and make products that are able to either suppress infections in farmed fish, also useful for human disease management treatment. So again, we call it from seed to society, and you can see marine biodiversity progressing through new bioactive agents to production, to application testing. And again, we're very careful at looking at societal aspects, and again, a large part of this is dedicated to the Bbnj process, the CBD process, and intellectual property management during the project. So we have an enormous amount of data this time. And again, at this point in time, it's important to recognize that that data has many databases that it can easily go. And again, the recommendation from the EU is that we use a sort of open data format for all of this. And all of this data, anything from the metagenome data to the bioactivity data to the chemistry data, the metabolomics data, and the compound characterization, all goes into open access databases that we can share and that everybody else can build on. My own work package, the one that I'm in charge of, again, does a couple of things. It looks for chemistry and identifies its chemical structure. Secondly, it tries to connect the genome and the metabolome. So typically, you go from genome to transcriptome to proteome to metabolome. But if we can connect the genome and the metabolome together, we can identify what the diversity is in the organism that might be expressible. And on the other side, we can go from the chemistry and go back to the genome. And again, that will be wonderful if that could be achieved in at least a group of molecules that we know very well, called the ribosomal peptides. The last thing of this project to do is to generate analogues using synthetic biology processes. And again, that's with a group of Jan Thiel and my own research group. And we try and work ways to make unique, what we would call unnatural products using engineered enzymes and engineered organisms. So synthetic biology approaches to make new molecules that would be able to treat disease. Screening and validation, again, carried out mainly at Medina in Spain and Icons in Italy. And again, we use libraries, we test them, we optimize, and we validate in that way what we're doing. What's really important for this project is that we have a very clear data management plan. That's insisted upon by the EU right now, and it means that the data that we get is reusable. Again, that was one of the biggest problems that the EU had previously, was that the data that was generated in projects often wasn't reusable by the next project. And again, this move towards open data benefits everyone. And the principles that Muriel spoke to yesterday is that we need to make the data findable, including provisions for metadata, that we need to be able to share the data, we need to make it interoperable, we need to be able to make it reusable through licenses if necessary in this case, because it might sometimes not be proprietary data from the project at the very beginning, and something that considers data security, ethics, and confidentiality. And we've already decided at this point which data repositories we'll use. And again, we've already seen the INSDC databases that we're going to use for genomic data. Metagenomic data will be stored in a magnified sub-database of DBI. Structural data, again, the chemistry structural information landscape is still not as clear as the biologists because there are several competing databases. So at the moment, we will commit to delivering it to one of these databases once we have looked at which one is the most appropriate for our particular subset of data. And then some data that, for instance, when we look at data and we specifically analyze it, that information will go into MyBig. Now, the most important thing here is that scientists are getting better at all of this, but they may need support. So projects like this should really think about investing in a data scientist in projects to be sure that that data is reusable in the right format for the future. We also have these work packages on sustainability, society, and valorization. And again, it just says to bridge the gap between marbles and the stakeholders, so industry, policy, and society. The environmental impact is in there, societal values, access and benefit sharing, and biosafety regulation. Link up policy with science-based contribution to maximize the potential for societal and commercial valorization. Again, a lot of this is very important to a project of this size. Again, this project is 8 million euros. And finally, we have dissemination, IP protection, knowledge management, and transfer and stakeholder engagement. Again, this is critical to for the European citizens to be able to establish the value or understand the value of the projects that we're doing. And again, for a project as interesting as this, it's not difficult to get public interest into what we do. I would like to just finish off now by talking about what's next. So a lot of these projects function very well and deliver data, but there's always the next step. There's the next request from the EU for funding, or they might have a group of interested scientists together. And this project, EU HotBio, is a European doctoral training network, was funded, led by University of Tromsø in Norway, the Arctic University. And the idea there is that we go further along the drug discovery pipeline, provide training for the next generation of researchers. And in fact, what we've done with this one is to encourage the previous project scientists to act as mentors for the current project students, in addition to which we offer the students opportunity to go to countries which are resource limited. In this case, Ghana and India have agreed to work with us. And it means that students can get an experience of, for instance, looking at antiparasitic agents in Ghana. Secondly, the Blue Remediomics project was just funded. And again, it focuses much more on tailored bioinformatics for the marine microbiome. It's a huge project led by the EBI in Cambridge. And one of the emphases on this project as well, apart from developing products and processes, is to develop an ocean microbiome health index. And again, this could be very vital to the kind of work that we're talking about in conserving a healthy ocean. And finally, only just, we heard about this just recently, it's a UK-funded project called the Bbnj project with the Natural History Museum of London, together with University of Strathclyde and the National Oceanographic Centre in Southampton. And it's a partnership with the Pacific Small Island Developing States, versus via the University of the South Pacific and the Cook Islands. Again, the idea here is to try and understand the value of biodiversity in deep sea regions at risk from mining and climate change. Again, so thank you all very much for listening. I hope that was of interest. This is the Marbles project, and we hope to meet in person now in Leiden in October. Thank you very much.
Thank you, Marcel. Thank you for bringing the biodiscovery process to life. It was wonderful to see the progress, I guess, from collection to finished product. Some of the things that I guess I took from that was the long road from collection to, you know, a finished product, which often takes around 20 years or so. You mentioned that you need a very large sample, you know, a really large number of organisms to have a chance of finding a promising lead. And I thought it was quite interesting that even though 55% came from the deep sea, you were saying that there were no leads that actually came from Bbnj. And I guess you also emphasized that nothing is unique really unique globally. I wonder if we could, I can't see any, oh no, there are some questions in the chat. I'll just reading in the chat. Hello, pleased to meet you. I'm very, oh, hang on. Well, if you're asking a question in the chat, would you like to turn your microphone off and ask in person? The person in charge of the APA mission? Is it Janine Fouché?
I think she wanted just to introduce herself. It's in French.
Sorry.
No,
Yeah.
don't worry. It's in French.
She didn't want to ask. Oh, introducing us. Oh, sorry.
Thank you.
No worries. So I'll just give a moment for anyone to turn their microphone off and ask a question. Otherwise, I've got some burning questions myself. Well, how about I just get the ball rolling and then we'll see if anyone else has some questions. I think, Ariana, did you want to keep this Q&A session to about 5 minutes, did you say?
Yes, if it's possible.
Yeah. Okay,
We have
no worries. Well, I guess to Yvonne, you were talking about the blast process a bit earlier, and you know, I guess when you were talking about it, you were talking about it's essentially a comparison with all of the other, you know, digital sequences in the database. Can you perhaps just talk a little bit more about that in terms of, I guess, in terms of the idea of, is this considered to be access or is it use of the sequences? And because in the draft text, you know, the language is around access or utilization. And so can you give everybody a sense, I guess, of just how many sequences are compared and whether it's active, kind of the idea of access?
Okay, the idea of access. Of course, I'm accessing the databases and I'm obtaining information from there. Legally, how you define access and how you use this definition of access to legislate on that afterwards. I mean, it's not something that I am here to tell about. But in order to produce this presentation, in which I have invested 10 minutes of talking time, I may have used myself sequences that I have produced myself 3, and I have compared them with all the sequence information available in the databases. So all the trillions of sequences that are there have helped me to tell in one circumstance which is the sequence that I had from my organism of interest. Otherwise, I wouldn't be able to know what ATGGCCCATG means. For that, I need the sequence information available in the databases, and whenever any of the sequences is retrieved, the information that I will be obtaining is not going to be so accurate. And on the other case, I did have the sequence information for a gene that I know because I was looking for it. I was looking for this gene because it's present in all the living organisms. And once I obtain it, I want to know this sequence to which organism it belongs. And through comparison with the databases, with all the information that is available in the database, I have been able to know that it's a copepod. And it's a copepod that is not natural to my waters. It's natural to the waters of Japan. And I have been able to spot that this copepod, for the first time, has incorporated into our waters through the comparison with all the information. If, for instance, Japan would decide that the DSI from the genetic resources of Japan is not available in the databases, I would have never been able to identify my invasive species in my coastal waters.
Thanks, Yvonne. That's really good to, I guess, give everybody an insight into just how many sequences you are effectively accessing when you're comparing or when you're doing a blast search. So that's really...
I must tell you that the blast that I have done it real time with you in front. I have used my sequence and I have launched it against all the databases and it has been compared in the web. The other thing that it can be done is you can download all the information in the databases to your own server. So you can do in your own computer all this blast. This is something that you need to do when you have plenty of information. When you are working with one single sequence, you can use the web. Otherwise, you need to retrieve all this information, and this can be done.
Yes.
Can I add one little thing to that, Fran? The important thing to remember here is that many of the annotations, like Iban showed, the function of these genes has been determined through years and years and years of laboratory work, maybe millions of years together, right? So and the key is often that for marine science, we don't know enough about marine genes because there are some quite unique genes there. And again, laboratory work needs to be done to do that. So if we don't share the information, we lose the ability to compare. And somebody finds a new gene in the ocean, determines its function, uploads it to the database, and it's there for other people to compare it to. And that's critical. So again, I'm very glad that you mentioned you query the entire database. So in fact, every single sequence has made some sort of contribution to your final search.
Thanks, Marcel. And so I guess, Marcel, then talking about the biodiscovery process, so even going pre-DSI, so when you're talking about, can you explain a little bit more about, I guess, whether it's feasible to have, say, scientific identifiers to follow the sample from the point of, well, to have identifiers, follow from the point of collection all the way through the, DSI once it's dematerialized in the databases and then through to the finished product. Can you explain a little bit more how an identifier might work along, or if it may not work along that path?
Yeah, so I think Muriel explained it really well yesterday, but I'll try and sort of recap very briefly what she said. I mean, the key is that there is typically with a research vessel, you may have a cruise number. You will then use that number to identify a sampling event. A sampling event may well be something that you collected in a particular way. It comes to the surface, and that is then given a identifier on the vessel. When that specimen is deposited in the museum, let's say, or a collection, it's given this globally unique identifier that Muriel referred to yesterday, and that should then be cited whenever you deposit data elsewhere. So you might deposit data in the INSTC, you might deposit data in ChemSpider or NT-Atlas when you find a molecule. As long as that information is linked and it's basically machine readable, what could happen is those databases could be linked together, or basically the database could find that information for you. So again, we tried an example from the literature, and even with the most primitive identifier, we were able, because the French IFREMER deposited their sample data set and was findable with a very short code, we were able to use that to trace everything all the way from collection to DNA sequence deposit to publication to application. Unfortunately, when the product went into industrial development, it was no longer traceable. So again, there should be a way in which industry could share that information with the public databases, so that at least you know where to find it and who to ask.
Thanks, Marcel. I know we've only got maybe 2 minutes left, or maybe less. But I guess one of the other things that is in the draft text, in terms of one of the options, proposed options, relates to depositing the samples as well as the information in publicly accessible databases. How feasible is this based on your experience? So, could you just briefly explain how much of, say, the original sample is typically saved during the research and development process for a requirement to have that put in, say a duplicate sample in a database, in a collection?
Yeah, so it depends on what work you're doing with that. So often samples are sampled for particular reasons. So I remember a collection expedition to Fiji a long time ago. I was prepping samples in 7 different ways for different purposes downstream. However, if you want to look at a good example, Marbank, the Norwegian biobank, has a good process right now for people accessing samples. So the samples are deposited, they're recorded, you can find them online. And what you can do with that information is then say, well, I would like to access that with legal certainty. You can then write to them, and they'll say, what would you like to do with it? And what they say then is, well, because samples are limited, they have a kilo maybe of sample, they don't want to give it all away to one person, so they'll share 100 grams with you, as long as your work is not competitive with other work they've already done. So if you're going to test for cancer, and cancer has already been tested for in that extract organism, they probably wouldn't give it to you. So again, it's a very clear process. Again, it's allowing the maximal use of those samples with the minimal overlap of usage, so complementary usage. Again, that's the way I would see that possible. And again, in some cases, there may not be enough sample for any further work at all. So again, if it's a microbe that's isolated, then of course the microbe is culturable and could be in fact shared given the correct procedures.
Thank you, Marcel. And I know we've run out of time now, unfortunately, because there's so much more to talk about. But thank you again, Yvonne Cancio and Marcel Jaspaz, for very enlightening presentations on the biodiscovery process. Thank you so much. Thank you, Ariana.
Thanks, Fran. Thanks for staying up late.
And thank you, Fran, for moderating very well. And we invite you to stay on if you manage for the Q&A question panel later. So now we go to panel 6, capacity building needs and ongoing programs, where I will be the moderator because unfortunately Salah Tajikabi Bau is not attending today for health issues. And sorry, I apologize. I forgot in the beginning to introduce myself for the one that didn't attend yesterday. So I'm Arianna Brogiato and I'm part of the Bbnj EU negotiating team for Bbnj. and I'm focusing on MGR, of course. So I will introduce the first panelist of this session, which is Lucy Woodall. Lucy Woodall is a marine conservation ecologist. She is a scientist at the NetCon and an associate professor at the University of Oxford. Her PhD was on the conservation genetics of European seahorses, and she now seeks to understand the patterns of biological life in the deeper waters and the consequence of human activities on them. Lucy is featured as one of the Inverse Future 50 profiling people who will shape the coming decade. And she is co-lead of a DOSI Marine Debris Working Group. Lucy is also a co-founder of the Heritage Marine Foundation. So please, Lucy, you have the floor.
Great, thank you so much, Ariana. Let me just share my screen here. Okay, let's hope you can see that. Good afternoon, everybody. Good morning and good evening, depending where we are across the globe. I've been asked to share a few reflections on the deep sea expeditions that I'm involved in. And these expeditions are co-created by partners. I'm going to talk today about an expedition with Seychelles and Nekton, and it's Nekton that I'm a principal scientist for. In these expeditions, we combine both capacity building, or as I actually prefer the term, knowledge exchange, when we combine this with science and with communications. And all of these parts were really interlinked. So let's just move on here. So you're not just going to hear from me, you'll also hear from Sheena Talma on the left, who at the time was working for the Ministry of Agriculture, Climate Change and Environment in Seychelles, and also Stephanie Marie on the right, who was working for Seychelles Fishing Authority, both of whom benefited from the Seychelles Climate Change and Adaptation Trust and Nekton grants. And they were able to join the expedition to take part in all of the activities, but also critically to be able to lead their own research. So as you might imagine, in the deep sea, for our research, there's many people involved. So I did want to start this presentation rather than end by it, just sharing some of the 40 different partners who are involved with all the logos there, and some of the over 150 people who were involved and really essential to the project. And this picture, in fact, was taken at the end of the expedition, and we have the ex-president of the Seychelles who joined us, President Danny Fall. So Seychelles is in the Indian Ocean. It's a large archipelago. It's over 99% ocean. It has a rich history in coastal and shallow water science, and that is represented by the two logos of some of the many organizations, the Seychelles Island Foundation and Nature Seychelles, who have provided a lot of information about those shallow waters. Seychelles also committed to 30% protection of their marine environment in 2012. And it is some of the information that these organizations that I've just mentioned and others put towards the Marine Spatial Plan that was conducted. So a comprehensive marine spatial plan facilitated by TNC started in 2014. In 2016, the first Debt for Nature swap that was broken by TNC occurred. And then after multiple multi-sectorial consultations, and this is a lot of interviews with different members of the stakeholder community, then 30% of Seychelles waters were gazetted in March 2020. And these areas are represented here on the slide in green, the Zone 1, which is the higher protection area in dark green, and the multi-use area in light green. So it was off the basis of this that we conducted the expeditions to the Amarantes Islands, which are there in the middle with the smaller green, dark green areas, and also out to the Aldabra group, comprising Aldabra and Astove as well. that we were working on. So I'm just going to share sort of the different phases in the expedition. I'll show a few examples after this slide of the different activities, but I'm not in this presentation going to be able to touch on all of the different aspects. So phase 1 was establishing partnerships and planning. And then we moved into phase 2, which was the field phase. And of course, this is very obvious and very clear, but we can't do this without really establishing those partnerships and planning process. Phase 3 was about the workshops for exchanging our knowledge, for sharing new knowledge and being able to collect that information. With phase 4 being around the stakeholder discussions about the consequences of our new findings, about co-disseminating that information as publications and other outputs, and then critically being able to deposit that information and curate the samples, as we've just heard about yesterday and from Marcel in the last talk. So just a few examples here. This was one of the planning sessions where we discussed the process of going to sea, making sure we aligned our timelines, our science objectives, and where we wanted to focus our work. And on this workshop, we agreed on where the knowledge gaps were from across all of the stakeholders and the partners in Seychelles. And this really used a lot of the learnings from the marine spatial planning process and the additional management needs that had been highlighted through that process. So this is a very integral part of other activities that were being conducted in Seychelles. I'm next going to share just a few slides from our expedition in Seychelles, because obviously, we're talking about the ocean, but we've seen very little of the ocean in our slides thus far. So I just wanted to share with you the boat, the Ocean Zephyr, that we used. She wasn't a science boat, but we mobilized her with 18 different technologies on board. And one of those technologies was a submersible, and we used this to be able to visualize the sea floor. It also allowed us to be able to take policymakers and politicians down to experience their own waters. So that this submersible really had multiple different rationales for use. So now I want to share Sheena's thoughts, as I promised that she would be.
There is no audio to this.
Okay. Apologies. I'm sorry that there's no audio. That must be a problem with technology my end. I really apologize. But in the interest of time, I think I'm just going to move on. The next slide was actually with lovely Stephanie Marie telling us about her experience about going into the submersible. But I think because there's no sound, I'm going to move on. But I did want to tell you about Steph. Steph, on International Women's Day in 2019, became the first ever Seychelles aquanaut when she went down to 250 meters and was the very first person to ever see that bit of our planet. And she tells us very eloquently about her experiences, but hopefully I can share those with you at a link at a later date. So in phase 3, I explained that we went and did workshops. These are taxonomic workshops. This was the addition to the Seychelles coral collection, and you'll see the extra specimens being presented there on the slide. We conducted visits across our different scientists, exchanges, and we created networks of shared knowledge as well as training, symposiums. We also were able to co-disseminate some of our research, and we'll see a picture of Sheena and I there on the right when we were talking in Marlborough House at the Secretariat for the Commonwealth in London. And of course, this work is still going on. And the final stage was that we're in right now, and that's curating and depositing the specimens. So the specimens are going to go on loan to the Museum of Western Australia, until facilities permit curation in Seychelles. However, all of the data are currently held in Seychelles and now are being ready to be shared on open access databases like those that we have presented here on the slides. And they will have their internationally recognized identifiers. So really talks back to what Muriel and Marcel have already shared with us. But importantly, I wanted to share just now in the last few slides, just some of the reflections on the importance of having co-design and co-development as part of our knowledge exchange offering that really goes alongside the science. So because we're able to do this together, we're able to decrease the time that it took for decision making. We created future plans for the development of these plans. And I'm just going to share with you some words from the Ministry of Climate Change and the Environment in Seychelles. And they said that understanding the collaborative process was also a valuable legacy for Seychelles. This expedition and associated activities provided an indication of how programs can be run and where opportunities might be available for those Seychellois researchers who are interested in ocean science. Following this work, standards for engagement have now been established by the ministry. So that's a really powerful statement about the value of this type of work. And we're also able to set up different templates and ways of working to ensure we had high communications. In the future, we want to do more peer-to-peer learning. And in fact, I'm delighted to share that we're co-creating an expedition with Maldives partners currently. And this is a large focus, with over half of the scientists on the expedition from Maldives and the majority of them from across the Western Indian Ocean region. Also, regarding technology transfer, the submersible that I showed you is just not possible for all of us to use at all times, but we can combine that with low-cost, high-tech technologies that we're trialing on this next expedition. So I just wanted to leave you with some take-home messages. That co-production was not only the right thing to do ethically, but it also achieved more and better outcomes and critically outputs. So in this final slide, I'm just sharing with you a link to the paper that we wrote on this. I did try and stick to the 10 minutes, so hopefully I'm not too far over there, Gianna. You can go to the paper if you want to find out any more, but I'm also in Lisbon, so is my email address, and please do come and we can arrange a meeting. I'm really delighted to share anything that might be of interest to you. So thank you so much for your time.
Thank you very much, Lucy. That was really interesting to see. And I would love, as a policymaker, to be taken on ship or on ship. It would be really nice. So I introduce now the next speaker. So the next speaker is Dr. Sara Swaminin. She's a marine molecular ecologist working with the Ocean Biodiversity Information System, Odis, of IUCN UNESCO, on project for the applied use of environmental DNA for invasive species management, the PACMAN project, and monitoring marine biodiversity, eDNA expeditions. She has a background in marine microbiology and received her PhD from the Royal Netherlands Institute for Sea Research. She's passionate about enhancing the efficient sharing of genetic biodiversity data and how it can inform future development in marine science and policy fields. And she is presenting with the Ward Appleton, who is the Marine Biodiversity Focal Point of UNESCO Intergovernmental Oceanographic Commission, where he managed the Odis and support the biology and ecosystem panel of the Global Ocean Observing System. So I leave the floor to Sara. Thank you.
Hi, everyone, and thank you so much for having me. I'm having a bit of a trouble sharing my screen, but I'm asking Ward, he can maybe share it for me. So I'm here with Ward Appleton, who is the project manager of Odis. So he will also be available for chats later on and in the forum. Let me just see. It's saying that I don't have the permission and I have to restart if I want to share.
I can make you, just leave me a second. I will make you host, so hopefully you can present because I don't have your presentation. So just a second and it will be possible, I think. Change role. I'll make you co-host. It should be okay.
Okay, just a second.
Let's try.
I think it's on my side. Ward, are you able to share?
I have the same problem. I also need to quit and reopen, which I can do, of course.
Okay, maybe. Did you send your presentation to me? I don't think
No.
Okay, so in the meantime, you can send it to me maybe, and we can go to the next speaker. So then I will download it and I will project it for you. So the next speaker is Lucy Scott. And she's a marine scientist with over 20 years experience in 12 different countries, mainly in the African region on large marine ecosystem, GIS atlas and data and information projects. She has a master's degree in ichthyology, and has previously been the science and data manager on the Agulhas and Somali Currents LME project. She is currently project manager of the IOC UNESCO Ocean Info Hub project. So if Lucy Scott is with us, I hope you can share your screen.
Thank you. Thank you very much. And thank you for the opportunity to join. I'm also trying to share my screen.
Okay,
It's telling me to,
it's
telling me to quit PowerPoint and start it again. So I'm just doing that now and I'll be able to share in 1 minute.
Otherwise, I have your presentation, so.
Great. I should be able to.
If Lucy also has to quit, maybe I can share my screen now. I think I'm ready.
It keeps saying open system preferences, so I'm not winning here.
Okay, I can show your presentation, Lucy. Okay.
Thank you.
Let me just open it. It happens. I mean, we are used to this kind of hiccups. Sorry, everyone, but I guess it's normal.
Thanks so much.
Let me just open it. Should be this one, no?
Yes, thank you very much.
Screen?
Yes.
Can you see my screen? It's in full. Okay, now should be in full screen. You have the floor and you tell me when to change slide.
Perfect. Thank you very much. And thank you again for the opportunity to join this important webinar. My name is Lucy Scott, and I'm project manager of the IOC Ocean Info Hub project. Is my sound all right? Thanks, you can proceed.
Sorry, I think I need to change something in my audio. Sorry for this. Sure. Content and only automatic. Now it should be better.
Okay. Thank you very much. So this first slide is just a quick introduction to the Intergovernmental Oceanographic Commission. It's a functionally autonomous part of UNESCO, founded in 1960 with 150 member states. The scope is international cooperation and coordination of ocean research, services, and capacity building. Next slide, I'll go through these introductions. There are a number of IOC regions and also thematic project offices around the world, as well as a number of global functions, including ocean research, observing system and data management, early warning and services, assessment and information for policy, sustainable management and governance, and capacity building. Next slide, please. So the IOC of UNESCO supports a number of services which facilitate access to data and information. Next slide. The one that I'll be talking about is the Ocean Info Hub project and Ocean Data and Information System. And my colleagues Ward and Sara will be talking about the Ocean Biodiversity Information System. So the Ocean Info Hub project aims to improve global and equitable access to ocean information, meta, data, and knowledge products for management and sustainable development. The project leverages a digital exchange architecture to power an openly accessible web platform supporting communication and interoperability between distributed and independent digital systems. So it's an interoperability project, and not setting up a new centralized resource, but linking together existing resources. The project is funded by the Government of Flanders, Kingdom of Belgium, for 4 years. Next slide, please.
Sorry, it doesn't work anymore. What's happening now?
So the project has supported the development of the Ocean Data and Information System architecture using common data exchange conventions such as schema.org, which are well supported to facilitate this interoperability. Next. And the project is first working with IOC associated online resources. And we can run through the next slides quite quickly because it just names the resources we're working with first, which are Ocean Expert, Aqueducts, the Ocean Best Practices System, the Ocean Biodiversity Information System, which we'll hear about just now, the World Ocean Database, and a number of other resources that, such as Eurocean, marinetraining.eu, EMODNet, and other sources in the IOC Odis catalogue of sources. Next slide. We're working with 3 pilot regions, the Africa region, Latin America and Caribbean region, and the Pacific Small Island Developing States. The project benefits marine and coastal stakeholders around the globe, but its initial focus is on these 3 regions. They requested these services initially, and we've been working on a process of co-development with communities of practice in these 3 regions. Next slide. Together with these communities of practice, we identified initial data types which were of highest importance, and these include experts and institutions, documents, spatial data and maps, research vessels, education and training opportunities, and projects. Next. And so we've worked with our partners to develop the Ocean Data and Information System architecture. It's been developed, tested, and fully documented, although it is an ongoing process, so we continue to improve the documentation. And we're working with a number of partners that are already indexed in the knowledge graph, including the Ocean Best Practices System, OBIS, Ocean Expert, Aqueducts, the Western Indian Ocean's Maspawira project, and the Marine Institute. And we're working with a number of other partners that are online independently, and we're working on developing their linkages. In total, we're working with, at the moment, over 30 implementation partners so far in a process of co-design. Next slide. We've developed a reference implementation of the global hub architecture to demonstrate the search facility across the existing working implementations, so to show that it works, proof of concept. And we're currently in development of a front-end global search hub, also as a demonstration of this architecture. Next slide. And the global demonstration might provide tools such as finding specific expertise, finding vessel-based training opportunities, peer-to-peer collaboration, finding data or information, and finding spatial data or metadata. Next slide. The benefits of Odis to providers and users include to users. There are many portals for many kinds of users that serve up information in the way that they find it easy to access and use. For contributors, they're able to share metadata only once to retain ownership and control of their own data and expose only what they choose to share. And for partners, anyone can access the knowledge graph and develop their own portals for thematic or regions of interest. We're developing the infrastructure and proof of concept, and we're making it freely available. Next slide. So how the Ocean Info Hub project might support access to marine genetic resources, data, and information is that obviously this initiative supports the FAIR principles, most importantly by making information more easily findable, but also accessible, improving interoperability and thus the reusability of information if it's well described. Next slide. And specifically, the Ocean Info Hub and Odis architecture might improve discovery of information about the occurrence and distribution of marine biodiversity in Bbnjs, connectivity between species and ecosystems, human uses of Bbnjs, and area-based management tools in Bbnjs. So thus, the Ocean Info Hub can facilitate improving the availability of data and information on marine genetic resources, creating greater interoperability or facilitating interoperability between databases of importance, and making information on other aspects such as EIAs or patents that relate to marine genetic resources from Bbnjs. Next slide. Finally, this is a closing slide just to show some of our documentation. Our technical documentation, as I mentioned, is open and freely available. We also have a training course resource online. This was held in October, but resources remain online and openly available as a demonstration on how to link systems into Odis and make them a more visible and actionable part of the ocean's digital ecosystem. Thank you very much.
Thank you very, very much. So we can now go hopefully to Sara and Word and their presentation.
Yes, hello. Thank you again.
Can you try to project your presentation, please?
Yes. So Ward, can you share the presentation? Great.
Otherwise, I received it and I can do it. Okay.
Okay, it's working now. Great. So thank you again so much for having us. So I am the scientific officer for OBIS, the Ocean Biodiversity Information System. And as I said, my colleague Ward, who is the project manager, is also here today. And I'm going to tell you about what OBIS does and also how we are developing local and global capacity in biodiversity data management and also in monitoring using molecular methods. So what OBIS does is one of the largest marine biodiversity databases in the world with free, accessible, and detailed marine data. And so currently we have over 100 million presence records of species with over 182 million measurements and facts. So these are, for example, environmental measurements or metadata related to sampling, which is in connection to the presence records. These records are coming from 4,500 data sets with 164,000 different species. So the data is stored in community data standards called the Darwin Core, which we also heard mentioned earlier. And this makes our data interoperable with other biodiversity databases as well as machine readable. So in this map, you can see all the occurrences we currently have across the globe. Of course, we have more information in coastal areas where more of the work is being done, but as you can see, we do have data sets from all over the globe, also in Bbnj. So in the next slide, you can see we have selected all the Bbnj data. So it is actually 8.6 million records with 27,700 marine species from approximately 1,200 data sets. And so this data set, you can very easily visualize and download it via this web address here. So everything is openly accessible to anyone who is interested in it. So the next slide, please. So OBIS originates from the Census of Marine Life. This started was a project in 2000 to 2010. And the ultimate vision of OBIS was that you could have an individual selecting any area or volume of water on a global map and bring up information on what has been reported to live there. So since then, we have been steadily growing. We were adopted by the IOC UNESCO as part of the IOD program in 2009. And in 2019, if you show the next slide, please. At the 30th Assembly of IOC, the 150 member states recognized OBIS as a global open access data and information clearinghouse on ocean biodiversity for science, conservation, and sustainable development. So next slide, please. So as I said, we have a wealth of data about various marine species, habitats, and whole ecosystems. But through OBIS, you can also see how this data is being used. We have, for example, a list of the publications that are citing OBIS on our website, and you can see that last year this was already 180 publications. And these publications are from all over the globe. So this is a map of the authors of the publications and specifically the connections between the co-authors of each of these papers. So we had in total over 1,800 research publications from scientists from over 73 countries. And this graph, you can see that, yes, the north-north collaborations are still stronger, though there are also quite a bit of north-south and south-south collaborations, though these can still be improved. So we think that this metric could be something that could be used to monitor improvements in benefit sharing and international collaboration. However, we think that this map really shows that through open access to data, OBIS provides equitable access and benefits to research and enhances international collaboration. In addition, OBIS supports several global and regional ocean assessments with our data, for example, the UN Second World Ocean Assessment. And these reports and assessments are, of course, used to enable science-based decision-making. But OBIS is not only data. We have extensive international networks with collaboration. Our data comes from geographic and thematic nodes. We have over about 32 nodes, which actually are providing all the data. We are also in collaboration with many international initiatives like GeoBon, MBON, and Goose. And in addition, we promote a culture of cooperation by supporting training and capacity development. You can see we collaborate with the Goose, so the Global Ocean Observing System, to develop a global marine biological observing system and essential ocean variables that would measure the state of the ocean and different indicators. So next slide, please. So in the next weeks, we are launching an important new tool to support this work, which is the Biological Observations Monitoring Facility. And this tool will record the information on long-term sustained biological observation programs. So it details the information that they are collecting, where and in what frequency. So there will be links to this data and protocols, as well as who to contact if you want to get involved in any of these projects. Next, please. So a very important part of OBIS, as I said, is capacity development. We provide training in data management and biodiversity, in the use of Darwin Core standards, in quality control, data publishing, data access, and now, of course, also in capacity development for genetic methods. Most of our training, or all of our training, is done through OTGA, the Ocean Teacher Global Academy. This is a global network of 17 regional and specialized training centers that provide both in-person and online trainings. We've already organized more than 20 training courses with over 300 participants. And now we also have financial support from Norway to develop a fully fledged OBIS training course with online tutorials, so training can be done online in a self-paced mode. In addition to training courses, the European Commission has also provided important financial contributions, which have allowed our OBIS nodes to organize grants that support data owners to digitize historical data sets. And then what about genetic data? So what we think we believe is that this genetic data that now can be collected, it has really enormous potential to increase our access to biodiversity information globally. So this can be environmental DNA, where you analyze the DNA from simple environmental samples like water samples to understand what species are close to that sample. Specimen DNA, like we've heard from some of the talks during this webinars, or bulk sample DNA. The challenge we have now is how can we make this DNA data collection globally accessible to every part of the world, and that we make sure that we share this data in an open and reliable manner from the very beginning. Now, as we've heard already, quite a bit of data, so the raw sequence data is shared by most studies in databases connected to the INSDC. However, in many cases, this data does not allow to search for species in time and space. And this is because you are sharing raw data instead of your analyzed data, so your species list that you are getting out of your environmental DNA projects, for example. So now with OBIS and other biodiversity databases, we're accepting this genetic data, and what we really want is to have species information that is linked to sequences as well as spatial coordinates and time. And this will really allow the comparison with different data sets as well as different data types. So through OBIS trainings and projects in eDNA collection, we are really committed to mainstreaming genetic data collection globally, so making this accessible for everybody, as well as developing standard open access data management workflows that follow FAIR principles. And we have two ongoing projects. So one of the projects is the Pacific Islands Marine Bioinvasions Alert Network. The next slide, please. Where we have partnered with the University of South Pacific to develop a marine invasive species monitoring program with the use of environmental DNA sampling. And here you can see our Pac-Man project manager, Jope Ginigini, who unfortunately wasn't here today, and then processing samples at the USP laboratory. What we're doing in this project is developing it with strong local stakeholder involvement, sharing our training programs through the OTGA, having a sustainable data management workflow, as well as developing a decision support tool for early warnings. This monitoring program will be developed in a way that it will be a toolkit that then can be used in other areas of the globe as well. We have done the co-design of the monitoring plan with international scientists and local authorities. And everything was done online because of COVID. We were not able to have face-to-face meetings, or they were not able to have face-to-face meetings in Fiji either. But we managed, and we also got in-kind support from some key local stakeholders in Fiji, like the Biosecurity Authority of Fiji and Port Authority. The second project that we have is eDNA expeditions in marine World Heritage sites. This is a new project that has started at the beginning of this year, where we are partnering with the marine program of the World Heritage Center in developing citizen science sampling campaigns at marine World Heritage protected areas, where we aim at creating baseline information on the diversity of marine fish and endangered species in these sites. And why we were inspired to do this was really because the collection of eDNA samples is so simple that any person without extensive scientific training can do it. So we want to capitalize on that possibility and bring this sampling into the hands of the local community and citizen scientists and youth. So what our aim is to measure species diversity and to estimate possible changes due to climate change, but especially to inspire and educate a new generation of marine scientists, to increase awareness of the importance of marine protected areas, showcase how sampling of eDNA can help the local community in protecting their area, and promote the open access of data. So that's it on what OBIS is doing at the moment with genetic data and capacity building. And in conclusion, we are an established database acting as a clearinghouse mechanism for all different data related to marine biodiversity. The technology and methodology for data integration is already in place with a network of over 1,000 institutions and 32 nodes. We collect.
I think we lost you, but right at the end. I don't know if Ward want to add something to this last slide, or am I frozen? Can you hear me?
I can hear
yes,
you.
Ariana, I don't have any problems.
Maybe I can just quickly finish.
Yes.
I think this is really the last slide, so I'll just quickly finish. So we collect the genetic data with all kinds of metadata. So this will allow comparisons throughout time and space and across different data types. And we are developing local capacity in molecular methods. And through this, our goal is to mainstream biodiversity monitoring globally and offer full open access workflows. So next slide, please. For more information, you can find the potential IUC contributions to the Bbnj agreement in this paper, which was published in September 2020. And that's it. This is a picture of all of us working at the Secretariat. Thank you very much for your attention. And I would also like to thank all of our sponsors to the OBIS Secretariat. So thank you.
So thank you very much to all the panelists of this panel. And I have to apologize, but my computer got frozen and I needed some help and I couldn't unmute myself. So this is why I created dedicate 5 minutes of question to these specific panels, and I have a couple of questions to kick off, and then we can go to the Q&A panel discussion with all the experts. So I noticed from
do you want to keep the weekly with Rich
from
on
Lucy
Tuesday,
intervention, you make a reference to how the Ocean Info Hub could be useful for MGR of the Bbnj.
Yeah, I guess you can reschedule it. I mean, it's fine to reschedule it.
And you made a reference to making information on patent available, which really attracted a bit my attention. So I would like to ask you to elaborate a bit on this point, which seemed to be very interesting to me. Can you please mute yourself while you are not talking? Thank you.
I didn't hear all of the question, but I think I got the gist of it. Thank you very much. That was one example, just showing a type of information that might be useful to be discovered. So it's not something we're working on at the moment, but it was just an example of how it might be useful to the Secretariat to access patents or to access marine genetic resources, just to say that that could be a category of information that could be discovered through the Odis architecture and the Ocean Info Hub project, if that facility were to be developed. Thank you.
Thank you very much. I would be interested to know more from this in the future, so we can keep in touch by email maybe. And I would have another question to all the three panelists, which is, as we did last time, we tried to get away from the expert. What suggestion would you feel to make to the Bbnj negotiator from your point of view, from your intervention today? What the Bbnj agreement should ensure to facilitate your job and the value that your job is promoting, so enhancing scientific knowledge and capacity building. Who would like to start first?
So I can, as I spoke first in this session. There we go. Then it makes it easier for everyone else. I would say that I think negotiators would be really wise if they were thinking about authentic interactions. We've seen that there are new opportunities coming through for groups to be able to network, but actually to be able to create networks that have trust and have transparency across participants is really challenging, and this can only be done with time. So I would say really think about authentic interactions to make sure that networks and relationships that are developed are actually meaningful.
If I can add to this, Ariane,
Of course.
Yeah, I think, yes, I want to second this. And I mean, we've seen very nice examples from the Necton project in the Seychelles and the Pacman project in Fiji, and now the expeditions we're doing in 25 UNESCO World Heritage Sites. It's really engaging with the local communities. And we also saw the presentations from yesterday that I mean, it's really important that that we increase the capacity of doing science in all countries, including developing countries. So I really hope that this is something that Bbnj can achieve. And we saw the presentation on Odis with Sara, showing all the connections of researchers working together on publishing papers. I mean, if we can improve that figure and get a stronger connection with the South and also with South countries, because there were, I mean, it looked impressive, but there are still a lot of countries missing from that map. That shows that even if we provide open access to data, it doesn't mean everybody has the capacity to actually use the data and do science on it. So that's building the capacity locally is something that we really need to do.
Thank you very much. Can I react a bit provocatively to this, which is a very important point, but I'm just considering I'm the Bbnj treaty, so I am a treaty, and of course I can provide the framework to enable some action, some further capacity building, but I think maybe my question would be what I could do because it's more IOC who is doing the project on capacity building. What could the Bbnj do for your job to be easier, let's say? Because I don't think a treaty can really deliver in itself the action. It can facilitate, of course, and provide the structure, the legal structure to facilitate it, but the action is more for the entities that are already in place, hopefully, and then I see other people have questions.
If I can just quickly react on that one. So IUC is indeed, I mean, is an organization that can support UNCLOS in its activities, but IUC does not have. I mean, we don't have anything that we decide is not legally binding, and so I think it's important that Bbnj creates a framework and some of the principles that then upon the activities should happen, or Pierluigi yesterday also said, like, if we focus on the data and define on what the data requirements and the metadata requirements are, I mean, the same can happen for capacity development. What are the needs, what are the requirements, and what are the principles that we should operate on? Then, I mean, that if there is a legal framework, then it's also very clear to all the parties on how things should operate, and then we can operate in that space and facilitate and support.
Thank you very, very much. So I see Laurie from UK wants to ask a question, and then I saw also Chris Bowler asking for the floor. Laurie, you have the floor.
Many thanks, Ariana, and many thanks to the presenters and all of the presenters again this afternoon. Just a question. I mean, we've just had amazing presentations of some really amazing work that is being done. One of my takeaways from it, however, is and this is no criticism at all, they're brilliant projects, however, they do seem to be very focused in areas within national jurisdiction. I just wondered whether the panel had any thoughts about whether there are particular challenges we're doing work in areas beyond national jurisdiction, we need to bear in mind either in the negotiating of the treaty or maybe more relevantly in its future implementation. Thank you.
I think that's a very good question, but I also see a hand raised by Chris, who has some expertise also in areas beyond national jurisdiction. So maybe.
Thanks, Ward. I hope you can hear me. Yeah, I just wanted to chime in and say that, yeah, not always the Bbnj and the coastal areas are nicely separated from each other, because oftentimes, particularly when we're talking about DSI, we're talking about sequences that have been, we're talking about information that has been reconstructed from both samples taken from Bbnj and samples taken from somebody's national waters. Okay, so perhaps there needs to be some thought about, yeah, how do we deal with that? You know, when the DSI that somebody may be wanting to exploit is actually derived from, you know, a combination of different areas. So just a spanner in the works for the lawyers there. That's all.
Thank you very much appreciated to have more tasks. Anybody else wants to contribute on this point?
I would just like to say, yeah, I completely agree. I mean, biology doesn't sit within specific limits. So we don't have those clearly demarcated areas when we're thinking about going to sea. And as someone who does conduct research in the high seas as well as in national jurisdictions, I think it's very unusual that I will see completely different types of organisms in the high seas. So there are challenges, of course, when we go into the high seas. Areas are more remote, but I do think that the examples we've seen are actually very relevant to the future work that we're all doing. And in fact, yeah, high seas work is being planned in that area. So yeah, I can update you on that, Laurie.
Thank you very much. And I saw Christina having her hand raised for a moment. Okay, she's posting a link to the webinar that just was held Monday, I think, on connectivity, which is available. Okay, so unless, do you want to add something on the webinar, Christina? You're free to say if there was any good takeaway for this.
Sure, I'll just sort of say that because these ecosystems are so interlinked within and across boundaries, deep sea to the coastal zones, as Anna Metaxas stressed in the IUCN webinar, it's if we want to really adopt an ecosystem approach to managing resources or species beyond national jurisdiction, it would really help to provide developing countries, however you wish to describe them, make sure everybody has the capacity to actually get to know their own backyard, and their own backyard may extend well beyond national jurisdiction because of those very connectivities that we're talking about. So looking at a regional perspective, trying to get an idea of the biogeography of the regions or the ocean systems, as Simi Payne likes to call them, would basically start to provide really good grounds for trying to join up a lot of these capacity building efforts, the technology efforts, to be able to understand what's going on within and beyond national jurisdiction, because they are all interlinked. Over to you, Ward.
No, I would just want to add that indeed, when you can do training even in coastal areas and learn the job, you don't always need to go into the high seas. Actually, I mean, I was thinking out loud, coming back to the fact that doesn't make sense to limit ourselves to areas beyond national jurisdiction is I would assume setting up an agreement in areas beyond national jurisdiction might actually be easier to start the whole process. To start than going into national jurisdictions, because things might become a lot more complex, and not every country will agree on setting a law that affects activities within their jurisdiction. So, in a way, if we can have an agreement on how we can work together in terms of doing science and training and developing the knowledge that we need to manage our ocean, if we can all consider the areas beyond national jurisdiction as our joint laboratory, I think that could be a very good start. And actually, at the legal point, maybe an interesting way to start collaborating together.
Thank you very much. We have another question from Fran. You have a floor, please. Thank you, Fran, for staying with us.
Thank you. This is a question for Sara. In the draft text, it seems to assume that humans are collecting the materials and from areas beyond national jurisdiction, like in Article 9, it talks about the scope relating to activities carried out by natural persons. I was just wondering if you could give the room any idea of whether fully autonomous vessels are already collecting eDNA from areas beyond national jurisdiction and how that would work. So if it goes from, the information goes straight through to databases without effectively being carried out by natural persons.
Yeah, thanks for the question. I might give the word to Ward, who may have some more information on this, but just wanted to note that in our capacity development, we're really trying to have that people can collect their own samples. So to have very simple protocols for local people to collect their own samples. But of course, for autonomous sampling, Ward, do you have something to add?
Yes, there are a number of institutions developing these devices to collect eDNA samples with underwater robots afloat. To be honest, I don't know yet if they are operational in areas beyond national jurisdiction. But yeah, I was wondering, what is the question exactly is?
It's more about, I guess, future-proofing the draft text, because if the draft text is focused on the current technologies, with humans collecting the samples, or and if this is on the horizon or if it's already happening, is there a need for negotiators to think about, new technologies and making the language in the text accommodate that?
Yeah, well, the robots will take the samples, but the samples will still be treated on land, and in the lab. So it's not that I don't think the DNA extraction itself and then the sequencing will be done by the robots in the ocean. So it's just a matter of how it's just a collecting device. It's still the samples are still a property of the country or the institution that is running the robot. So in that way, I don't see a big difference. Yeah, Chris, you have your hand.
Yeah,
yes,
please. It's beginning to happen, and the technology is ramping up pretty quickly. So we will definitely see more and more of it in the near future, I would say. Probably within 10 years or so, we certainly will have the capacity to sample, extract DNA, and do sequencing in the middle of the ocean. It's not going to be tomorrow, but probably in 10 years' time, we'll be doing it. Yeah, it's the technology moves very quickly, so it would be wise to consider that. Yeah, that would be my opinion.
Thank you. Can I just react to this, to you and Fran? Because we were also reflecting on this, and we came to the conclusion that apart from, samples landing into a lab and being analyzed by humans, I think, but maybe I'm wrong, so I stand to be corrected. Also, the devices are sent and organized by institutions, so by humans. Even the collection part is still under the responsibility of an institution, of a research institution. So yes, so maybe we don't really need to change that much the language, because there will still be a responsible institution within a state sending out this robot to samples, but maybe I'm just wrong.
Yeah, no, that's right. And in terms of the flag state would be responsible in that respect. But I guess, in terms of AI and its legal kind of personality in many countries is not necessarily certain yet. So if there's, in terms of the nature of the technology, it's dealt with in different ways in different countries. But, as to whether a state would necessarily have jurisdiction over AI itself, in the sense of, that obviously jurisdictions have, states have jurisdiction over the activities of their natural persons and judicial sort of bodies. But I suppose the point is that it's relatively unclear in terms of the legal status of artificial intelligence. And, I guess having neutral language in the text could head off any kind of argument that those kinds of activities wouldn't apply.
Thank you very much for the suggestion, and I guess Laurie want to react on this point, so I give you the floor, Laurie.
Yeah, thanks, Ariana. I hope you don't mind me coming in again, but just from the perspective of someone who works with running our existing marine scientific research program, the kind of permitting program in the UK, and has an interest, and we work closely with our scientific institutions to look at what developments are coming down the track, how do they fit within the UNCLOS framework. And certainly from a UK perspective, the existing kind of remotely operated kits or the gliders, we do stick a Union Jack on the side, and kind of assert the kind of flag state approach in that way. So if they are coming from our research pool, they've got a Union Jack, we would say that that's under our jurisdictional control. I will say, I think we do think the language natural or juridical person gets quite difficult and may cause more problems down the line. And jurisdictional control may work better in those circumstances. Thanks.
Thank you very much. I see also in the chat, Christina is asking, but what if a research vessel is government owned and operated? Can you elaborate a bit more on this, Christina? What do you see as a difference?
Hi, and maybe
I'm not sure I understand the question.
Question is maybe for Laurie, and if you don't mind my putting you on the spot, is that Article 3 sometimes seems a little bit overbroad. My understanding is this language was intended for MARPOL, that was states that may not need to apply the MARPOL requirements, but are still obligated to do their best to do so. But this Article 3 actually exempts any vessels owned and operated by a state and used only on government non-commercial service. And many research vessels are government owned, from US NOAA vessels to UK. So I'm just curious, what are the implications of this article for research cruises that go out to that include sampling of marine genetic resources as part of the processes? It's not relevant here, but I think it will be relevant in the long term. Thank you.
Ariane, if you're happy, I can give a kind of view from the UK. Yeah, it is a tricky one. It is language that is very important to the UK in terms of certain vessels that are government owned or operated a non-commercial service. And of course, the language itself is taken from UNCLOS. But however, we do see the issue with what we don't want to do, certainly as the UK, is carve out state-owned research vessels from the marine genetic resource part of the agreement. So we are sort of looking at this language, working behind the scenes with other interested states to try and find if there is a way of finessing this that would bring clarity to it. But just to be super clear, from our perspective, we had that it wouldn't work if we didn't apply it to the. So we will be applying it to our government-owned research vessels. The question is whether there's a way of finessing that language that works for everybody. Thanks.
Thank you very much. So I guess we are already in the panel number 7 on debate with all the experts. I see most of them are already here, and we already kicked off with very wide questions. So I would just leave the ball rolling and see if anybody has a question on all the different panels that you listen to today, and you listen through my summing up in the beginning of today from yesterday. I think what was important in this flow of different panel is to go from certain very important elements related to the MGR chapter. So the focus on conservation of biodiversity hotspot we had yesterday, and the link with the CBD convention, and from where we started a very interesting discussion on DSI. We wanted to underline that we are perfectly aware that the DSI issues in Bbnj has not been, sliced as much as in CBD, and there is basically no agreement between delegation on this topic, and also on the benefits derived from DSI. But we anyway wanted to hear what are the discussion in CBD to get a flavor of why they are shifting from a bilateral approach based on track and trace, and they are shifting towards an approach which is more multilateral, it seems, at least in some of the options. And here, why they are doing this, because track and trace is, as I repeated today from their words, not feasible technically and also economically, probably. And we heard also from the very nice work on the DSI that we had yesterday and today, but even scientifically it doesn't make sense. We heard this from Tara yesterday, that the value of the DSI is the comparison between DSI coming from different jurisdictions and different locations. So this is why we are convinced that a different view at these issues will be needed one day in the Bbnj discussions. So yes, I would really encourage people not to be shy and profit of all the experts that are here with us today, or also the expert if I have any comments that might trigger other questions. I leave the floor open. Please put your name in the chat or raise the hands and I will look for you. Or if you want to put the question in an anonymous way, just put it in the chat only to me, to my WebEx account. Okay, so I see Amber, she wants to intervene. You have the floor. Thank you very much for breaking the ice.
Yeah, thanks. No, it's been a really interesting, and Fran might have had her hand up at the before I did, but nevertheless, it's been really interesting coming more from the CBD world to listen to the conversations that you've had over the last 2 days. I think it does sort of create in me a bit of deja vu with our discussions in the CBD space around tracking and tracing compared to where we are now. And as you rightly pointed out, Ariana, I think that there has been, through many nights and mornings of online webinars, because our intersessional period went from being 2 years to 4 years. And we have had the time to, I think, discuss and look at the technical angles a bit more. And although I wouldn't say that the idea of tracking and tracing is completely gone from discussions in the CBD around DSI, I think that there has been a realism about the billions and the trillions problem, right? And the slide that I had to skip yesterday that I'm almost tired of saying, but it's 280 million sequences when it comes to the CBD scope, 1.5 trillion reads, the entire data set, all 280 million, are downloaded to 34 million times a year by 10 to 15 million users. And then they're propagated through hundreds of thousands of publications and patent applications. And so if you sort of wrap your head around the combinatory problem of millions of sequences downloaded millions of times by millions of users, thinking about a tracking and tracing, your head just sort of blows up. And so I think where the conversation has evolved, and maybe this is useful, and we've heard, just really incredible examples of how the ocean science community has done a lot of efforts around tracking and tracing. One might be tempted to think that the scientific system to keep track of data could be used as a regulatory mechanism. But I don't necessarily think that that's a one-to-one relationship. I think that what we've been talking about in the CBD discussions is that you can use scientific databases, scientific tracking and tracing systems to add an element of transparency in terms of reporting, in terms of indicators, in terms of checking in, is this system working like we planned it to be? Is it functioning? But you don't necessarily have to turn to scientific systems to become regulatory police actors, because that's neither what they were built for, nor necessarily creates the right incentives. And so if you wanted one person's opinion, I would just say, treasure your scientific integrity and reproducibility and the transparency you've heard about today, but don't let it fool you that it is your regulatory system. My opinion.
Thank you very much. This is a very powerful message, and I think very important, because the science and policy interface is here for this reason, because we talk different languages. You're scientists, biologists, bioinformatics, and we are more lawyers, policy people, and we need to understand each other. And when lawyers look at your subject, we try to assimilate through our concept, and our concept is a checkpoint, a legal number that tells me something, because this is my room of comfort. So this is where it gets really difficult to translate your own language into something that works for me, but still work for you. So I'm not translating it in something that is different. I think this is the science policy interface that we need in these elements when things get really, really complicated. So thank you very much for your very frank and upfront suggestions and also lessons learned of deja vu. Do we have, yes, I see Fran. She has a question. You have the floor, Fran. Thank you.
I've got a question for Lucy Woodall. I was just wondering, based on your experiences of your field trips, do you think it would be fairly straightforward to have pre and post notifications for those collection expeditions as proposed in the draft text?
Yeah, that's a really good question, Fran. I think what would be possible is suggesting where you're likely to be going and what you're likely to be focusing on. I think in reality, we know that putting on expeditions is really expensive. So if the weather's bad in one place, you're going to go somewhere else. If you see this amazing thing that you've never seen before, then you'll want to go and collect it, as long as it's not the only one in that environment, right? So I think that there does need to be some flexibility within the text to permit that kind of curiosity-driven science from not being removed from the scenario. So I think it's totally possible, but within that remit of flexibility.
Thanks.
Thank you very much. Can I pick up on this and ask you, and also Chris Bowler, and if you see in the pre-cruise notification enough room to integrate opportunities for developing countries scientists to be on board of a research project, not only on board of a ship, but in a collaborative way, like Lucy described, from the beginning of a project to the publication, hopefully. Do you see a pre-cruise notification, if done well in advance, as, you know, a hotspot for advertising that these opportunities are there and that there are funding and there are spaces? Or do you see it as difficult to be implemented and to get good results? Because we don't want to do PR, we really want to create opportunities. Thank you.
Well, I will start, and I'm really interested to hear what others think, but I think that it potentially
Anybody
can
wants
be
to
used
try to answer?
as an advertisement. But I think that for me, I would find that really challenging, because I co-produce science. That is part of my research philosophy. So our research questions, and hence how we go about our science, what equipment we use, where we go, what time of year we go, is very embedded in that. And that comes down to the scientists with whom we're working. So if I was to put on an expedition and say, oh, I've got a berth, come along, that's not an inclusive way of doing this. And this is what I meant by meaningful interactions. That's an interaction. Is it meaningful? Well, maybe or maybe not. But certainly to get the most out of a process, I very much believe, and we have proved it in our work, that really scientists are going to be involved from the very beginning through to the very end. While none of us might be perfect at doing that, I think if there's that ambition, then that is really important. And I do worry when I see the opportunities for berths to be available, to really ensure those scientists are empowered to do things that they're interested in, and that everybody's got that open dialogue. So I don't think it's impossible, but I do think that there's much more meaningful interactions that happen if there's a long-term process.
And what would be your suggestion to ensure that this long-term process is in place?
Well, I guess that is the $1 million, €1 million, £1 million, and every other currency question, isn't it? And I think part of it comes from maybe a change within the community of understanding how we conduct our research and valuing different types of knowledge, and valuing different types of expertise and experiences, and seeing where that value does magnify. But yeah, there needs to be additional consideration of that, because I don't think just having a berth available permits there to be a fully interactive and positive experience, because scientists who do not take part in creating an expedition will be find it much harder, I think, to be able to fully interact. And also, it doesn't give them the opportunity to be able to lead their own parts of the projects, which is as important as participation. And that's how we're going to be able to really get those global views within a global representation that is meaningful.
Thank you. Before leaving the floor to Chris Bowler, maybe I would like to add another question on this bit. Do you think that the funding agency would maybe be better placed to kind of ensure this long-term process you're referring to by incentivizing at the start when you settle down with scientists and you decide where to go, what to focus on, as you said, and if you have certain requirements put by the funding agency, maybe this would be better?
I think that's a really interesting suggestion. I think that funding agencies have an important part to play. They have an important part both for what they insist on within grants, because let's face it, it's from our funding agencies where our money comes from and essentially will determine what we do. But I think it's also for them to better understand the process of really valued co-production and co-design. They're very, relatively small words, but actually they mean a lot. And that process of really conducting research and the timelines involved are often quite different than some funding bodies currently acknowledge. So I think there's an education piece and there's maybe an implementation as well. Yeah.
Thank you very much for sharing your experience. And I leave the floor to Chris, which I'm sure will have something to contribute on this topic.
Yeah, I think Lucy already made some very valid points. Perhaps to add to what she said, I think the funding agencies are more and more aware that we need to engage internationally, and I see more and more often, applications for cruise time on a ship or application for research projects. The funding organizations have to have a component of having one or two or several berths available for people beyond the project from developing countries and so on. So those sorts of mechanisms begin to be operational in several national funding programs that I see more and more. So it begins to start. And I think that's a very positive step to see that. In terms of notifying pre-cruise, I don't see a major problem for scientists, but I don't think it will necessarily help to recruit interested scientists from elsewhere. Very often, we scientists we already establish our international collaborations over the long term. We have particular engagements with specific countries to try and help, to try and promote benefit sharing and so on. So oftentimes, we already have mechanisms in place to bring people on board from particular areas. So I don't think we necessarily need to advertise berths that are available, because you have these we need to have long-term engagements with particular organizations in other countries, and to assure that the people coming on board have a valuable experience and contribute to the whole thing and so on. So just the example of Tara. Tara is currently in off the West African coast, off Angola and Namibia, and we have local scientists on board. Yes, it's challenging to bring those local scientists on board. It's a challenge because of language from the start, scientific training on board. These are all, challenges that you have to face, and we do that in Tara as part of our long-term commitment with the Global South to promote benefit sharing, and it's very worthwhile to do it. It's very satisfying, and it's something that we will continue to do. So I hope that feeds a little bit the discussion around this topic.
Thank you very much, Chris. Yes, this is something we have reflected upon a long time, to how to better ensure these opportunities are there and are transparent and available and advertised everywhere. Probably we need to reflect a bit more on the long-term stability of these opportunities to be offered. And let's see what we can come up as language in the treaty to offer this, which is a very important point. But I feel share a comment or have another questions on today's or yesterday's presentation on what we have said so far. Let's see, is anybody asking for the floor? I also put in the chat the list of participants. If you can fill in with your details, please. Yes, I see Ward has a question. You have the floor, please. Thank you very much.
Thank you, Ariane. I want to bring up one other discussion point, and that's the making the samples and data available in publicly open access databases. What the
Are you there?
Yes. Can you hear me, Chris? Yeah, you can hear me. Okay. So what it doesn't mention is the information derived from the data and the samples. So what we often see is what's happening is, I mean, GenBank is a huge database and that has a lot of advantages, but there is also a lot of disadvantages that sometimes people just dump the data there without very good quality control and not with all the information derived on sequences. So I think a lack of a clear open access data policy in Bbnj can actually hamper making progress in conservation, and that's because, I mean, what I now see is, I mean, there are a lot of sometimes commercial companies developing genetic reference databases because they are providing a service to monitoring biodiversity, but without the full transparency of what science, what open science actually means. And having, I mean, of course, having a very reliable and authoritative genetic reference database requires a lot of work, and there's a lot of intellectual property involved in that. But I kind of see a problem if we continue doing this and not making genetic reference libraries publicly available, because we will continue blasting our sequences to GenBank, which might not give us the best results compared to what the commercial companies can provide in terms of blasting capabilities and reference libraries. So I would strongly advocate for a full open access data policy, also for the information derived on the data, not just for the raw sequence data. I don't know if that's clear, my point.
Yes, thank you very much. I might ask you to elaborate a bit on this, because if I'm understanding correctly, you would wish either the Bbnj system or, policy makers to make open access every kind of databases, also private databases. Probably if I got right your suggestion, but I leave it to you, and then I see Guy want to intervene on this point.
Yeah, maybe Guy, if you want to react on this.
So yeah, so this is a really interesting point. Really, just to stress that so I agree with a chain of open. You know, there's a process here of going from very raw forms of data that are close to the machines, that actually they're not directly interpretable, through all sorts of processes to normalize, to do QC, to clean up, and to build references. And so that's a whole chain of events, and each step is really important. And the further the openness can go along that chain, the more powerful this is, and the more science will advance around the data. But I think one of the key elements one needs to deal with is that what is feeding the beginning of the chain has to be correctly described and provided with the appropriate context. And this is all about data standards and metadata standards and reporting standards. And I think that to build those, there's no one authority that can say these are the standards you have to comply with. I think it's a cultural it's a cultural frame of mind that a community can develop over time, where publishers, institutions, scientists, databases, and the legal side can come together to help to develop and instill the use of these standards. So I think all of these downstream things really become possible when you capture richly described and well-structured data, and that's an important basis for this chain. And then the onward value is there, that one can build these derived databases to continue the value chain.
Thank you very much. Sorry, I have a bad connection, so I didn't really grasp it all. But I guess it's only my problem. Do you experience bad connection, or you hear better?
I have a good connection.
Okay, so I see there is a question in which. Okay, thank you. So it's my problem. I see there is a question from Fran on the chat. I will read it for you. So, do any of the presenters have experience with how scientists manage access to traditional knowledge of indigenous people and local community associated with MGR or the Bbnj? Is mention of traditional knowledge included in metadata in sequence databases if it is related to the original MGR? That is a very interesting question. Thank you, Fran. Anybody wishing to share experience on this important topic?
So I can.
Yes, there is somebody saying yes. Guy, you have a floor.
I can have a go at commenting on that. So I think traditionally the sequence databases don't make any great attempt to capture traditional knowledge on marine genetic resources, but not on any genetic resources. There may be some embedded in there, but I think it's not an area in which the managers of the databases and the scientists that operate them can necessarily add huge value directly. So that's an area where we look to linking out to other databases and other data resources and sources of traditional knowledge, and that would be, I think that would be the most effective approach at having well organized traditional knowledge in its own domain by people who know how to structure and know how to present that, but in a way that's linked accurately to the sequence records themselves. And so certainly that's an approach actually the sequence databases take for a lot of data types that are not central to the sequencing data model. Rather than build a data structure and operate services for these what we'd consider to be external data types, we go to pieces of infrastructure and authority services that we can link to, and we work with those groups to build and maintain those links.
Thank you very much. And I see that Amber wants to intervene on this topic. You have the floor, please. Thank you.
Yeah, and I understand why Fran asked the question in the way she did with a focus on MGRs and Bbnj. But I would say again here, there is nothing special or different about traditional knowledge in the context of MGRs and Bbnj than there is from the rest of the world. And so again, here my plea would be as a scientist for harmonization. You know, when traditional knowledge is used, be it from Eskimos or from the middle of the Amazon, the point is traditional knowledge. The point is equity. And if there is a system to integrate and to link that data, then it can't just be special for MGR and then different or incompatible with other systems. So whatever we think about the solution, my plea would be to make sure that it's sort of integrated into the broader community, and ongoing movements and in other lanes of traffic, if you will.
Thank you very much. And yes, indeed.
Sorry, I was just going to ask Lucy. I was going to ask Lucy, when you're talking about, the sort of authentic interactions, do you have any kind of involvement when you've got people on board from, countries or maybe traditional owners who might be on board your vessels to incorporate the traditional knowledge aspects in the research project from the beginning? Like, have you come across any of those sorts of?
Yeah, thanks, Fran. So I haven't done that myself because I haven't really worked in those areas where that's appropriate. But for example, we do on expeditions both in South Africa and Comores, we've brought on board the local artisanal fisheries and spoken to the fishers about their knowledge. So it's a different type of knowledge, but again, it brings us all into that same space. And, it really is fantastic to be able to have them on board and get them to see the bits that they've, considered their backyard for all those years. But they can share with us so much knowledge as they're also learning from the imaging that we're providing. So I don't have anything direct to support with that, but yeah, if I was to work in those parts of the world, then that would be a definite advantage, and would seek to do that.
Thanks.
Thank you very much. I see we are running out of time. I would like to make just a final comment that what was made yesterday by Amber in the chat, which I found it very interesting. So during the walk on DSI, thanks to the presentation of Yvonne, Amber made us realize that when he was accessing all these databases and show the interoperability between one or the other and showing us how you do a blast, I learned this new word. He never had to register or to provide any information when accessing these databases. So this is another point we need to consider in the meaning of what is open access and open science, and the fact that databases are not tracking who is accessing them, and they are not tracking what scientists are doing. I hope I'm using the right word. I don't know if somebody wants to elaborate on this, but I think it's also an important things to underline from these 2 days. And thank you both to Yvonne for walking us through DSI and to Amber for underlining this point, which is also very important from the policy and law point of view. So I think unless anybody wants to add anything, I would like to thank you all very much for your active participation yesterday and today. It has been really a pleasure to be with you and to discuss things. Apologize for the little technological hiccups. But on behalf of the European Commission and IUCN, DOSI, and also the Marpol project, We thank you for your time, for your dedication to this policy science interface, which is very, very important for us. And we might get in touch again with you experts for other questions. And thank you very, very much. And to the delegates, we'll see you soon in other intersessional webinar or workshops, and we wish you a lovely evening, afternoon, or morning. Thank you again for being with us, and we would like to thank once again Ambassador Rina, who stayed with us, and Profit as well, as we did of this important, interesting presentation. Thank you very, very much.
Thank you.
Bye.
Thank you, everyone.
Bye-bye. Thanks all. Bye-bye.
Bye-bye. Thank you.
Thank you all. Bye-bye. Thanks so much.
Bye.